Hello dear viewers, myself Nasir and you're watching Mr.Zoologist YouTube channel. Today we are going to discuss about principles of inheritance and variation. So we are going to look about in detail. First of all, we are going to go through the important terms. What is genetics? Study of inheritance, heredity and variation of characters or study of genes and chromosomes. Means to say, when we study about how information transfers from parents to offspring and how variation changes occur in within the single population, these will be called as their study will be called as genetics. Next is inheritance. Inheritance means to say how characters are transferred from parents to offsprings or parents to progeny. It is the basis of heredity. Means to say how these characters are maintained is the inheritance pattern. Next is variation, difference between parents and offspring. When little bit changes comes in the generation, that will be called as variation. Next is character heritable feature among the parents and offspring. For example, eye color, height, and so many other traits, which are also called as characteristics. Variants of a character is called as trait. For example, brown eye, blue eye, red eye, black eye. These are traits. What are alleles? Alternative forms of a gene. For example, tall and dwarf, short and long. These are maybe the two alleles of a gene for that character height. Next are homozygous, the condition in which chromosome pair carries similar alleles of a gene. If they are similar, means tall to tall or small to small, they will be called as homozygous or true breeding alleles. Whereas heterozygous, the condition in which chromosome pair are dissimilar, contains dissimilar pairs of alleles of a gene. For example, tall and dwarf, long and short, blue and black. These will be called as heterozygous alleles. What do you mean by dominant character? The character which is expressed in heterozygous condition, which shows its features, is called as dominant character. It always indicates with the capital letter. Recessive character, the character which is suppressed in heterozygous condition, means to say which is not shown its properties, is called as heterozygous condition, your heterozygous character. It indicates with small letter, it always is written in the small letter. Whereas phenotype, physical expression of a character, means to say how it looks like, it will be called as phenotype, physically, externally. Whereas genotype, genetic constituent of a character, means to say either it is human, dog, cat, frog. These will be called as its genotypes. Whereas hybrid is a combination of two, an individual produced by mating of genetically unlike parents. When two different parents, when we can say that when two different species combine together or reproduce, they will produce a hybrid offspring. Gregor Johann Mendel was the father of inheritance. He has proposed laws in biology. These are called as Mendel's laws of inheritance. Mendel conducted some hybridization experiments on garden pea, whose scientific name is Pisum sativum. He experimented on them for about 7 years, from 1856 to 1863. What are the steps in making a cross pea in a cross in a pea? Selection. First of all, selection of two pea plants with contrasting characters. One character will be dominant, one will be recessive. Emasculation means to say removal of anthers of one plant to avoid self-pollination. This is female parent. Emasculation will always be done in the female parent. Next is pollination. Collection of pollen grains from the male parent and transferring to female parent is called as pollination. Pollination has two types, self-pollination and cross-pollination. If it occurs in the same plant, then will be called as self-pollination. If it occurs between two different plants, then it will be called as cross-pollination. Next, fourth step is collection and germination of seeds to produce offspring. Means to say seeds will germinate and will produce offsprings. What were the seven true breeding varieties selected by Mendel? They were stem height, which may be tall or dwarf. Tall will be dominant, dwarf will be recessive. Flower position, axial or terminal. Axial will be dominant, terminal last while will be the terminal. Whereas flower color can be white or can be violet. Violet is dominant, white is recessive. Pod shape may be inflated, means swollen will be dominant. Constricted Holland will be recessive. Pod color can be green, dominant, recessive will be yellow. Seed shape can be round, dominant. If it will be wrinkled, that will be recessive. Seed color, if it is yellow, it will be dominant. If it is green, then it will be recessive. What was monohybrid cross? A cross involving two plants differing in one character pair. They will be called as monohybrid cross. If we talk about only length, that will be tall or dwarf, that will be monohybrid. If we talk about only one color, that may be yellow or green in a single plant or two plants, then it will be called as monohybrid cross. Mono means one, hybrid means combination of two different plants. So that will be called as monohybrid cross. Mendel crossed tall and dwarf pea plants to study the inheritance of one gene. He took two plants. One was homozygous tall, containing capital alleles. One was heterozygous dwarf, containing a small alleles TT. From there, one gametes comes around and fertilization occurs. And during fertilization, first filial generation formed is heterozygous tall, T capital, T small. That was tall, but that was heterozygous, not homozygous. When he self-fertilized F1 generation, first filial generation, he got Punnett scare in which a checkerboard applied. He got 3:1, that was phenotypic, whereas genotypic was 1:2:1. 1 ratio means one was true breeding tall, whereas two were heterozygous tall, whereas one was homozygous dwarf. That was the results that he obtained. One was homozygous tall, two were heterozygous tall, one was homozygous dwarf. That is the genotypic ratio, whereas phenotypic was 3:1. Physically, three were tall and one was dwarf. Inheritance of one gene, the F1 when self-pollinated produces gametes T capital and T small in equal proportion. During fertilization, pollen grains of T have 50% chance to pollinate eggs of the T capital and T small. Also, the pollen grains of genotype T small t have a 50% chance to pollinate eggs of T capital and T small. Means to say, in the first filial generation, chances for both alleles capital T and small t were equal, 50-50%. 1/4 of the random fertilization leads to homozygous tall. 1/2 means to say 2/4 of the random fertilization leads to heterozygous tall, T capital, T small. 1/4 of the random fertilization leads to dwarf homozygous, T into T. If we represent it in the form of binomial expression, then we can say that half chances 1/2 were for capital T, half chances 1/2 were for the small t. Then we will scale both of them, and scaling will result into two equal equations that will result again into the 1/4% of tall true breeding homozygous, half percent were chances of the heterozygous tall, and 1/4% were the chances of the dwarf homozygous. Mendel self-pollinated the F2 plants. He found that dwarf F2 plants continue to generate dwarf plants in even F3 and F4 generations. He concluded that genotype of the dwarfs was homozygous. Mendel's for checking these traits were okay or not with the experiment, he devised different types of cross. First was back cross and then was test cross. Back cross between a hybrid and its any parent. That will be used to check either the ratios are okay or not. Whereas test crossing of organism with dominant phenotype to a recessive individual. As you can see that recessive individual was cross hybridized with the heterozygous tall hybrid. He got 1:1 for the test cross. Test cross is used to find out the unknown genotype of a character. For example, we have a homozygous recessive. We will when we will fertilize it into the dominant phenotype, then all flowers will be violet. As a result, when white was cross fertilized with pink, we will see flowers of violet color. Whereas half white and half violet were formed when unknown flower is heterozygous. When unknown flower is homozygous dominant, then all flowers will be violet. So we can conclude that a test cross can be used to determine what will be the genotype of the F2 generation. First law of dominance. Characters are controlled by discrete units called factors. Means to say these genes, either tall or small, tall or dwarf, can control the characters. Factors occur in pairs. Genes, because always genes are in the form of pairs, so characters will also, or factors will also in the form of pairs. In a dissimilar pair of factors, one member of the pair is dominant. Definitely, when a pair of different or heterozygotes are present, then one will be dominant, capital, and small will be the recessive. What does the second law states, which is law of segregation during gamete formation? The factors, alleles of a character pair present in parents segregate from each other such that a gamete receives only one of the two factors. Means to say, each daughter receives one allele from the parents. Homozygous parent produces two similar gametes. Heterozygous parent produces two kinds of gametes, each having one allele with equal proportion. Means to say, alleles distribute independently from each other and transfer to the offspring. If we talk about dihybrid cross, as we have studied earlier that monohybrid cross, it is a cross between two parents differing in two pairs of contrasting characters. Means to say, now we will discuss two characters at a time. So, for example, cross between pea plant with round shaped and yellow colored seeds with wrinkled shaped and green colored seeds, and we will get a dihybrid cross. When we brought about homozygous round yellow with the homozygous wrinkled green, then we will get heterozygous round yellow first filial generation. When we self-fertilized first filial generation, we get ratio of 9:3 ratio 1.